Hi, my name's Nechama, and we're going to go through the BIOPAC Muscle Physiology Lab. The first thing you're going to do is double-click on My Computer, and once that comes up, you're going to double-click on Bio201TA Network, and then you'll see something called BSL Lessons 3.7.6. Double-click on that, and the student lab will come up. We're going to start with experiment 1, so click on Lesson 1, Electromyography EMG 1. Click OK, and make sure to make a file name that is unique to your group so you can find your data later. Press OK, and then we're ready to start the first experiment. At this point, the lead should be correctly attached to the participant's dominant forearm. And since the conditions vary greatly between experiments and even within one experiment, we're going to start out by calibrating. Calibrating will allow us to take the raw data and normalize it and rescale it to % of a reference value. In our case, the reference value will be the maximum voluntary contraction of the participant's dominant forearm. The way we're going to do this is by first clicking Calibrate, waiting for the calibration to start, having the participant clench their dominant forearm as hard as they can for a couple seconds. Then the participant will release their clench completely and wait for the calibration to end before moving on. And it'll look something like this. So click Calibrate, wait for it to start, clench with dominant forearm as hard as you can for 2 seconds, and then release completely until calibration ends. Now that you're done calibrating, you can either click Redo Calibration if you need to, or you can click Record to start the actual experiment. You should understand that once you click Record, the experiment will start recording automatically, so I suggest that you watch the video before clicking Record so you know what to do. Once you click Record, it'll start recording automatically, and what you'll do is the lead should already be attached to the participant's dominant forearm, and the participant will wait 2 seconds according to the x-axis and then clench their dominant forearm weakly for 2 seconds, then release for 2 seconds, clench their dominant forearm a little stronger for 2 seconds, release for 2 seconds, clench a little stronger for the third time for 2 seconds, release for 2 seconds, and then clench for the fourth, last, and strongest time for 2 seconds, and then release. And then you should click Suspend right away. And then it'll look something like this. Release for 2 seconds, clench dominant forearm for 2 seconds weakly, clench a second time for 2 seconds, release for 2 seconds, clench a third time, stronger for 2 seconds, release, and then clench a fourth, strongest time for 2 seconds, then release, and then click Suspend. And now you're ready to move on to the next part. Now that you've finished recording the data for the participant's dominant forearm, you're ready to start recording the participant's non-dominant forearm. Now that the recording is suspended, you should remove the leads from the participant's dominant forearm and place them in the correct places on the participant's non-dominant forearm. Don't click Stop or else you'll have to redo the whole experiment. And now that the leads are placed correctly on the participant's non-dominant forearm, you can click Resume to record the data for the non-dominant forearm. Once you click Resume, it'll start recording, and everything will be the same as it was in recording the dominant forearm, but the leads are just placed now on the non-dominant forearm. And so wait for 2 seconds, clench weakly with the non-dominant forearm for 2 seconds, release for 2 seconds, clench stronger a second time, release for 2 seconds, clench stronger a third time, release for 2 seconds, and clench the strongest fourth and last time with the participant's non-dominant forearm for 2 seconds, then release, and then right away click suspend. Now that you're done recording the data for the dominant and non-dominant forearms, you can start analyzing the data. To do this, you just press stop, and you'll be given the option of clicking yes. Click yes, and then you'll be given the opportunity to listen to your data. I don't suggest that you listen to the data because in the past, students have listened to their data and then lost their whole experiment and have had to start over or spend a long time searching the computer for their data. And since it's not required to listen to the data for your lab, I suggest you just move on and analyze the data. To do this, just click Done. And once you click Done, you're going to be given options. So click Analyze Current Data File, and then click OK, and now you're ready to analyze the data. The first step is to go to the right side of the screen and click on the I-beam, and now you're ready to highlight different parts of your data. The other thing you have to do is make sure that the channels are set correctly. So you're only going to be setting this set of channels, and the left hand smaller box should say 40, and the right hand box should say mean. And now you're ready to start analyzing your data. So you're just going to be highlighting the different segments of the clenches of both the non-dominant and the dominant forearms, and you're going to be able to read the millivolts value for each clench in the box where it says mean. And once you highlight all the clenches from both the dominant and non-dominant forearm, you should record each value for the clenches on your data sheet. Once you finish that, you're going to go ahead and highlight the tonus for each of the forearms. The tonus is the segment between each of the clenches, and it represents the resting state of the forearms between the clenches. And you're going to read the value in the same box for each of the tonuses for both of the forearms. And once you've done that, you're finished analyzing your data and you're done with the entire experiment.